This is the part 2 in which we will discuss about the prokaryotic cells. Prokaryotic cells are those cells, as we already discussed, in which the nucleus is not present. So nucleus is basically absent. Prokaryotic cells are represented like bacteria, blue-green algae, mycoplasma, which is the smallest cells, and pplo, which is a pleuropneumonia like organisms, and they are generally smaller, like bacteria is very small like amoeba, and multiply more rapidly than the eukaryotic, because eukaryotic cells takes place meiosis, mitosis, but these generally multiply. Prokaryotic cells, like, they multiply by binary fission, so rapidly multiply, and they may vary generally greatly in the shape and the size. For example, four basic types of bacteria, like basicle is a rod shape, so rod is like a long shape, coccus is like a spherical shape, and vibrio is like a comma shape, and spirillum is like a spiral, so these are a spirals, spring like shape. So the organization of prokaryotic cell is fundamentally similar, even in the prokaryotic exhibit a wide shape and the functions. So this is the diagram which is showing the difference comparison of the eukaryotic cells with the other organisms. Like, this is the typical eukaryotic cells. Eukaryotic cells in which the nucleus is present, and this is the cytoplasm, and these are some organelles, like we have mitochondria, we have these are like ribosomes, microbodies, and other other organelles as well. So these are the prokaryotics, like typical bacteria, this is the pplo, and the viruses, etc. Now the prokaryotes have a cell wall surrounded with the cell membrane, and this is the cell membrane, and the fluid, that is the this is a fluid which is the cytoplasm. So there is no well defined nucleus, because in prokaryotics there is no nucleus is present, and the genetic material is basically naked and not enveloped by the nuclear membrane. So nucleus is not present, so it is generally naked and very not enveloped by any nuclear membrane. And many bacteria have a smaller circular dna outside the genomic dna. These smaller DNA are called the plasmids. So smaller DNA are called the plasmids, and the plasmid DNA confers certain unique phenotype characters of such bacteria. One such characteristics is the resistance to antibiotics. In high classes, we will study plasmid DNA is used in the monitor bacterial transformation. This we'll discuss later. So, a specialized differentiated of cell membrane is called the mesosomes. Like, these are some of the mesosomes which is present, is characterized by the prokaryotics, and they are generally essential in foldings of the cell membrane. Discuss about the cell envelope and its modification. So most of the prokaryotic cells, particularly bacterial cells, have a this chemical complex cell involved, and the cell involved consists tightly wound layer structure, which is the outermost glycocalyx, followed by the cell wall. And you can see whatever I have underlined, that is important, and otherwise it is the not important. Now the procedure is that gram stain gram positive bacteria, and do not call the gram negative bacteria, because gram positive bacteria contain lipid layer and gram negative bacteria does not contain the lipid layer. This is the only difference you have to remember. And glycocalyx differs in the composition and thickness among the different bacteria, and it is loose sheath slim layer, and otherwise it is a very thick tough capsule. The cell determine the shape of the cells and provide a strong structure to prevent the bacterium from bursting or the coalescing. Plasma membrane is semi-permeable in nature. Semi-permeable means some of the substances will transfer, some of the substances will not transfer by the membrane. And a special membranous structure is called mesosome. We've already discussed mesosome is the infoldings, which is the extensions of the plasma membrane into the cell, and these extension in the form of vesicles, tubules, and the lamella, and they help in the cell formation and DNA replication. Like, the DNA replication means formation of the new DNA and distribution of the daughter cells, and they also help in the respiration. So these functions are important, like what is the functions, and the secretion process to increase the surface area in the plasma membrane, enzymatic content. So in some prokaryotes like the cyanobacteria, these are the other membranous extension into the cytoplasm, which is called the chromatophores, which contain pigments. So except mesosomes, another some other extensions are also present, which are called chromatophores, and that contain the pigments. Now, bacterial cell may be motile and the non-motile. Motile is like bacteria like the amoeba, that is a motile. And if motile, they may have a thin filamentous extensions from the cell wall, which is called flagella. Because if they are motile, that means there is a presence of flagella. Also in the paramecium is the example. And bacteria show large number and arrangement of the flagella. Black bacterial flagellum composed of three parts: filament, hook, and the basal body. And you can study beside flagella, pili and the fimbriae are also the surface structures of the bacteria, but do not play the role of the motility. And the pili are elongated tubular structures which do not made of the special protein, and the fimbriae are the small bristly like the structures spouting out of the cells. So this is all about the cell envelope and its modification. So we have studied this about the prokaryotic cells, and this is also the ribosomes and inclusion body. So in prokaryotes, ribosomes are associated with the plasma membrane of the cells, and they are about 15 nanometer by the 20 nanometer in size, and they are made up of two subunit, 50s subunit and 30s subunit. So in ribosomes in prokaryotes is made up of 50s and 30s. In eukaryotics, this is different. Units which is present together from 70 prokaryotic ribosomes, and ribosomes are the site of protein synthesis. Remember, this is a site of protein synthesis, so ribosome where protein synthesis takes place. And several ribosomes may attach to a single mRNA and form a chain called the polyribosomes and polysomes. And the ribosomes of a polysome translate the mRNA to proteins. Inclusion bodies. Inclusion body is the reserve material in the prokaryotic cells which are stored in the cytoplasm in the form of inclusion bodies, and they are not bound by any membrane system and live free in the cytoplasm. Example: phosphate granules, cycophycean granules, glycogen granules, and gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. So this is all about the prokaryotic cells in detail.